I'll just run right into it. So orphan diseases, they're given the name orphan for a reason. They were by and large intractable for a very long time. Part of the reason they were intractable is they are quite rare. The general definition is they have to be found in less than 1 in 2,000 people. Because they're quite rare, one, makes them difficult to diagnose. There's really no expert on any one orphan disease by and large at every medical institution going. And this leads to, and this is a quote from parents of a child with an orphan disease that was being diagnosed here at the IWK. We found ourselves in a very frustrating, lonely, and frightening situation, mostly because of the lack of knowledge around both diagnosing the disease and then the disease itself when it was diagnosed. So the IGNITE project was set up to, of course, try and make this go away. The thing about rare diseases is rare is really not so rare. There are over 7,000 orphan diseases. Approximately 80% are thought to be monogenic, which means they're single gene disorders that are inherited due to a defect in a single gene. That means, and in terms of rare is not so rare, 1 in 15 Canadians is actually born with an orphan disease. The reason that you don't see a lot of people wandering the streets with these orphan diseases is 90% have no treatment, 90% are life-limiting, and 75% affect children. So when you do the math there, that's the reason that, you know, a lot of these children don't make it into adulthood. So, but it is 1 in 15, so that is a ginormous number as far as patient care that's currently going wanting, as you can see with 90% having no treatment. So orphan disease diagnostic odyssey. So this is a problem with orphan diseases because they are so rare. From the time a patient sees their physician, usually a child, to the time that they're diagnosed, right now it takes about 5 to 7 years. So that's a long time to diagnose a disease, and the average direct cost for physician visits and tests comes in at around $30,000 per patient. So it's a fair amount of money and a fairly long time. And a lot of times, unfortunately, the diagnosis will happen after the patient has passed away. So these are not spectacularly good prognoses from the diagnostic front right now for these rare disorders. So the IGNITE project was a team of clinicians and scientists that, of course, wanted to try and ameliorate this problem as best as we could. So it was research that we wanted to begin with patients and end with patients and their families. So we set up a process. There was already a very robust gene discovery platform that was taking place here in the Maritimes, the Maritime Medical Genetics Center out of the IWK and associated scientists. And to that gene discovery platform, the IGNITE project added model systems. One of the nice things from a scientifically tractable point of view is single gene discoveries are very easy to model because you just have to make the same mutation in your favorite model organism and you pretty much replicate the disease you see in people most of the time. So it's very scientifically tractable to study. And then once you have a bunch of model systems, we want to take advantage of a bunch of new technologies, some of which I'll talk about later, that then allow you to use high-throughput genetics or high-throughput drug screens to come up with a drug target or a drug that we could then return to the patient as rapidly and as cost-effectively as possible. And I'll also touch on very briefly some of the ethical issues because a lot of the new technology for diagnosing diseases involves sequencing entire human genomes, which is a brand new technology. And once you sequence somebody's genome, that's really their barcode, right? So you'll know a lot about that person right away. There's a lot of ethical issues associated with knowing somebody's entire genome. So this is by and large the team. It's based out of Dalhousie University, this IGNITE project, with some input from McGill and UBC as well. And we've really got 4 scientific objectives. The first is to ascertain and characterize patients with orphan diseases. Many of these come through maritime medical genetics, but others are seen by specialized clinicians, primarily through Capital District Health and through IWK. And if they have suspected inherited disease, of course, you usually search for known genes that cause this disease. If they have one of the known genes, great. But if they don't have one of the known genes that cause the disease, then of course what you want to do is move into the research platform to discover the gene that's mutated in that particular person that's actually resulting in them inheriting this disease. With that information in hand, You can then make your favorite model organism mimic of the disease using relatively routine these days gene replacement technologies and everything from a yeast cell to human cell in culture to a zebrafish to an entire mouse. And then once you have a plethora of model organisms that replicate the phenotype you see in people, you can then use high throughput genetics or high throughput drug screens to actually then try and identify drug targets or drugs that could be ameliorated. So the process is relatively simple in what we're trying to do. There were a lot of genes, as I said, prior to the IGNITE project. There was a project called AMBI that was very good at discovering novel genes through patients admitted to the Kaplan District Health and IWK, and IGNITE itself is continuing that tradition of discovering novel genes that cause rare diseases for patients in the Maritimes. And of course, these are then published, and the rest of the world then searches for these same mutations, and of course they'll find these in patients all around the world. And there's ongoing gene discovery projects as well. So why is gene discovery important? Well, gene discovery automatically gives you a DNA test, as most of you people here know, for the disease. So if you have a DNA test, that will reduce the diagnostic odyssey time to a matter of a month or so rather than years on it. So that's already a cost savings and a time savings, and you'll get a much more accurate diagnosis because you know exactly which gene and how it's inherited. And when you know this, you can actually give more accurate genetic counseling to the patient and their families in terms of what this type of disease and the mutation that causes means. The other important thing from a scientific perspective is when you discover a gene, it gives you insight into the molecular basis of the disease. So when this process is screwed up, it leads to this phenotype for this disease. And once you have insight into the molecular basis of the disease, this is the very first step required, of course, to move forward to trying to find a cure for the disease. So why now? Orphan diseases were given the name orphan diseases for exactly that reason. It was very hard to identify novel genes, and it was then very difficult to study. Well, a primary driver for the recent uptick in the study of orphan diseases has been the fact that we can now sequence entire human genomes. And we can actually do it cheaply. So in 2000, the first human genome was sequenced. It took a decade to do and cost $3 billion. You can imagine we can't do that for every person. But in 2014, an entire human genome can be sequenced and more or less analyzed in about a week for $5,000. And if you do the exome, so that's the part that codes for proteins, you can actually sequence and analyze that in a day or two for about $1,000. So you can imagine in just over 15 years, we went from a decade and $3 billion to a couple days and $1,000 to actually be able to analyze the major part of the genome that encodes most of the offending mutations that result in rare inherited diseases. So it's been this cost reduction that's now allowed us to survey entire genomes very quickly and cheaply to find the offending mutations that result in inherited diseases. So this is why now orphan disease research is actually doable versus a decade ago. And this just shows the number of base pairs in billions from 1989 through, well, this is 2012, that have appeared in GenBank, which is where all DNA sequences get deposited by and large. And so I started as a shiny new assistant professor at 29 years old in 1996 at Dow, and this was where it was. And now this is where it is here. So in my career at Dalhousie, they went from a few million base pairs that had been known in sequence for every organism to 140 billion. So that's a lot more data you have to work with as well, right? And that's because the cost of sequencing genomes has decreased so much. And this has led to a rapid rise in novel gene discovery. So this is every quartile from May of 2009 up until April of 2013. And this is using whole exome sequencing, so next generation sequencing technology, to identify offending mutations that result in an orphan disease. And you can see back in 2010, there was very little, 5 in a quarter. Now they're discovering 120 every 3 months, and it's just going up farther and farther. So Why orphan diseases and why now? It's pretty clear that DNA sequencing technologies, because of their cost and the fact that you can survey an entire genome so quickly and rapidly, now allow you to survey piles of people relatively cheaply in terms of looking at their entire genome. So everybody who has this disease, you can sequence their entire genome or their entire exome and zero in on the offending mutations a heck of a lot quicker and faster and cheaper than ever before. So where do we stand now as far as the orphan disease process? Over 3,000 genes have been discovered for orphan diseases. As we know right now, there's about 25,000 genes in the human genome. They estimate that there's about 4,000 more novel gene discoveries to be made. So that way they'll come up with the 7,000 rare diseases that are predicted to be due to single gene mutations. They anticipate all will be discovered by 2020. So within 6 years, they'll be done. And this is the International Rare Disease Consortium. That's a consortium of primarily North American and European universities and private sector companies that are planning on having this finished by 2020. So once you've actually got all 7,000 genes discovered by 2020, which is 6 years from now, the clinical exome will become a reality. So that means when a patient walks into the doctor and you say, I think I have some inherited disease, you'll get your exome done. And you'll be finished, right? So the plan is within 4 or 5 years of 2020 and having all 7,000 genes discovered and essentially almost all offending mutations discovered, they'll have a clinical exome process in place by 2020 to 2025 so that you'll be able to diagnose rare diseases just by doing some of these exomes. So that's their plan for the International Rare Disease Consortium over the next decade. So it's really going to change state of the art when it comes to actually diagnosing genetic and inherited diseases. With this, of course, and this is a little movie, there's ethical issues. It's from a movie I like.
What can it mean? The ability to perfect the physical and mental characteristics of every unborn child.
Can you hear this?
In the not too distant future, our DNA will determine everything about us. A minute drop of blood, saliva, or a single hair determines where you can work, who you should marry, what you're capable of achieving. In a society where success is determined by science, divided by the standards of perfection, one man's only chance,
How do you expect to pull this off?
I don't know exactly, is to hide his own identity.
This is the last day that you're going to be you and I'm going to be me.
By borrowing someone else's.
Congratulations. What about the interview? That was it.
Do you think you'd be doing what you're doing if it wasn't for who you are, what you are?
I'm feeling like the girl in the false confesses playing somebody else's hand.
I've got my picture plastered up all over the place. They'll recognize me everywhere.
They'll recognize me. I don't recognize you. They won't believe that one of their elite can suck them all this time.
They're going to find me.
But in a place where any cell from any part of your body can be trained, how do you hide? Or when we all shed 500 million cells a day. Ethan Hawke.
Okay, we don't need to know who stars in it. But there are a slew of ethical issues that will accompany sequencing genomes. And part of the process is, when a baby's born, and right now they take a blood spot and they'll screen for maybe 20 different orphan diseases, depending on what hospital you're in, through doing a combination of genetic tests and biochemical tests. They'll be able to do all 7,000 when the baby's born in anywhere between 5 and 10 years from now, prognostically. So you'll know beforehand that this disease is coming down the tracks with a very high level of confidence. You'll also be able to know predispositions to certain diseases like Parkinson's and breast cancer and other things where there's single or double gene mutations where we know that you will be highly predisposed to getting these types of things. And people may choose then to do prognostic things prior to getting sick. They'll then be able to decide what to do about their health, rather than waiting for the symptoms to occur and then showing up to the doctor's office. So it's really going to change, I think, a lot of things about doctor-patient interactions moving forward over the next decade. And doctor-genetic counselor types of interactions and all sorts of interactions are going to be quite different 10 years from now compared to where they stand right now. And those ethical issues are going to have to be talked about as well, everything from insurance companies to do you tell the children, who gets to know, do the children get to know when they're born, or not when they're born, but when they can understand it? Do you tell the parents, can you do a checkbox, I want to know, I don't want to know? There's cost of the healthcare systems, if you can treat somebody beforehand versus waiting till they get sick. There's a lot of issues that are going to have to be brought into play here, economic and ethical, to actually decide what to do with this information once we actually have it. So it can be used for good, but it can also be used for evil, as you saw from the clip. So we'll have to see where things go from there as well. So they're hoping in a decade, a lot of the orphan diseases will be known. There'll be clinical exomes available and diagnostics for essentially all of them. That said, then what's the next grand challenge? If that one's going to be pretty much close to solved, the next grand challenge is trying to do something about these orphan diseases. As you said before, as I showed before, 90% currently have no treatment, 75% prevention. So this is the next grand challenge, is going to be what can we do about it now that we can diagnose them? And so this grand challenge is part of what Ignite's trying to accomplish. And that's one of the things. As soon as you identify a gene, if the function of the gene is known, it will give you some insight into what fundamental aspect of cell biology or biology gets screwed up to cause this disease. And in some cases, and I'll show an example of this later, there's a disease gene that's identified, but it has no known function. So the first thing you have to do is figure out what it does to get some molecular insight into the disease. Now, once you have that molecular insight in terms of what it does, you can then make your favorite model organism mimic. And as I said, the nice thing about monogenic diseases and with the modern technologies is you can flip pretty much any disease-causing gene or inactivate any gene you feel like in everything from a yeast cell to a human cell culture to a zebrafish to a mouse. It takes longer and gets more expensive as you work up the evolutionary chain, but it can be done. So once you have these model organism mimics, you can then study them, one, to learn more about the biology of the disease, because you can do a lot faster, of course, with cells in culture than you can in people. And of course, there's different ethical issues. You can go as nuts as you want, throw bleach on cells if there's no forms to fill out, things like this, versus actually having to work on patients themselves. So you can do things very quickly using cells, and then when you find something that looks like it's very interesting and could be ameliorative to a cell, you can then move through, and what we use here is the zebrafish models in collaboration with Jason Berman and his folks. And then if it looks right in a zebrafish, whether it's a drug target that we turn off that ameliorates the disease, or a drug itself that ameliorates the disease, if required, we can then move into the mouse model. And their mouse knockout consortium is currently knocking out every known gene in mice, and they're also flipping in, as they're discovered as fast as they can, every known missense mutation that results in an inherited disease, so that you'll have models for everything across the board. And so once you have these model systems in place, as I said, you can study them to learn more about the disease, but also do genetic screens, i.e. a gene you can knock down or knock up that's ameliorative, or a small molecule screen. Screen for drugs, either virtually or in reality using high throughput robotics, and we do that in collaboration with CDRD and BC to again find drugs that are ameliorative to these model organisms. So you need some criteria. Right now we're going to have 7,000, we've already got 3,000 genes. In the next 5 years we'll have 7,000 genes that when mutated cause an orphan disease. We can't study all 7,000 at once. So we need to whittle this down in terms of a funnel and say, Which ones are we going to pick first? And so the criteria we've chosen is, of course, the nature of the disease suggests opportunities for intervention. So there's a period of normalcy before the disease actually starts to kick in. There's already an absence of an effective or inexpensive therapy. This isn't a hard hurdle to leap because 90% have nothing already. If there is presence of a more common phenotypically similar disease that might benefit from the identified drug, that would be good. So some diseases that are genetic in origin, phenotypically and diagnostically, look very similar to diseases that are not genetic in origin. So a drug that treats the rare disease may possibly treat the other disease as well. The relative rarity of the conditions, all things being equal, work on a disease that affects 1 in 5,000 people versus 1 in 5 million people to start with. And again, for us, the evolutionary conservation allowing for the use of as many model organisms as we possibly can to throw at the disease all at once. So that's our criteria for moving down the pipeline. So I'm going to give you an example of a disease called congenital sideroblastic anemia. I'm going to briefly touch on the gene discovery. I had nothing to do with this. This was a gene discovery made by the AMG folks, which is the predecessor for Ignite run out of Delhousie University. This started with patients being admitted to the IWK Healthcare Center, and they went to the hematology oncology group there. And it was discovered that they had this rare disease called congenital sideroblastic anemia. There was a disease-causing gene known called ALAS2 that causes this disease. They did not have mutations in ALAS2. So they had some unknown genetic something going on that was resulting in them getting this disease. So it came across the road, and they wanted to discover the gene. So what is congenital sideroblastic anemia? Well, it's an anemia. It means that you are incapable, or not incapable, you do a very poor job of finding oxygen to hemoglobin and then circulating that oxygen throughout your body so that your body can actually be oxygenated. So your hemoglobin levels tend to be low. They tend to have tiny red blood cells that accumulate iron. So that's what these little spots are in these red blood cells. And your bone marrow shows that you have ring sideroblasts in your precursors to your red blood cells. The one known gene was ALAS2. It is known to be the first enzymatic step in heme/hemoglobin synthesis, which makes sense. Okay, so if you can't synthesize hemoglobin, you're going to get the same symptoms. These patients did not have ALAS2 mutations, so what was up there? So they did the standard genetic mapping and sequencing of these regions that were suspected to contain the mutation in the patients, and they ended up identifying the gene SLC25A38. This was published in Nature Genetics a few years ago now, that showed that mutations in this gene SLC25A38 also cause congenital sideroblastic anemia. It was not only present in the maritime Canadian folks that were the sort of probands for this study. It was found in a slew of other people who also have congenital sideroblastic anemia from around the world. And then when people have now done these studies en masse, mutations in SLC25A38 are the second most common cause of congenital sideroblastic anemia behind ALAS2 mutations. So now that we know another gene, mutations in SLC25A38 can cause congenital sideroblastic anemia, well, one, we've already got a DNA diagnostic. We know it's mode of inheritance, so that can already help with diagnosis and genetic counseling. Can this help us lead us towards a treatment? There was only one problem. The first big problem was the function of this gene was not known. It had a name, but they didn't know what it encoded. So a gene makes RNA, which makes a protein, and the protein is a little machine in your cell that does a job, right? They had no idea what the protein was or what its job was. They had some ideas based on looking at the amino acid sequence. So protein is a string of amino acids, and Dan Gaston, who was a postdoc working on this project, put together this phylogenetic tree. Turns out SLC25 is a member of about 41 different proteins, all called SLC25 something, because they all look very similar to each other. They all are transporters, so they take something from one side of a biological membrane and move it across to the other side of the biological membrane. Okay, good, so we've got a protein that's probably transporting something. What's it's transporting? Well, they grouped with amino acid carriers. So amino acids are amino acids. There's other things that these proteins can transport. But this guy looked like, by just gazing at its protein sequence, to be an amino acid transporter. So we said, okay, let's try and figure out exactly what the function of SLC25A38 is. And we went to the most rudimentary organism where it was most conserved, and this was a yeast, Saccharomyces cerevisiae. It's the same yeast you use to make bread and root beer. There's also a lot of genetic tools you can use to play with this yeast, and where things are highly conserved and heme synthesis is very highly conserved, it's a very fast, quick model organism used to try and track down what functions of specific genes are. So we started here. So what does it do? Well, the fact that ALA has 2 mutations result in an inability to make heme, or if you're a red blood cell, turn that heme into hemoglobin. We checked what happens if you inactivate, and this is the yeast homologue of SLC25A38 to heme levels in a yeast cell, and they're decreased by half. And then if you express the human protein in the yeast cell missing the human homologue, you restore heme levels back to normal. So this lets us know that HEM25 and SLC25A38 perform the same function. So this allows us to now work on a yeast cell lacking the HEM25 gene, and it's the equivalent of knocking out SLC25A38 in a higher organism such as a human. So the fact that we knew a few things about the SLC25 family members. One, they're localized to the mitochondria. And 2, our specific family member looks like an amino acid transporter. So if you look at how is heme or hemoglobin made, there's 3 places where amino acids actually take place, could have a role. Serine is brought into the mitochondria, converted to glycine. This is where ALAS2 resides, and glycine plus succinate away make this 5-ALA metabolite that then goes through this long, complicated pathway to make heme and hemoglobin. All right? Glycine can come directly into the mitochondria and be converted, or 5-ALA needs to be exported out of the mitochondria. So is this transporter a serine transporter, or a glycine transporter, or a 5-ALA transporter? That was our hypothesis. We thought, well, the most obvious thing would be to be a glycine transporter, because that's the actual substrate for the first reaction synthesis. So we tested this hypothesis. Can SLC25A38, or the yeast homologue HEM25, transport glycine into the mitochondria? The first thing we tried, which was dreadfully disappointing, because it didn't work. I've spent 20 years working on lipid metabolism and things that can transport stuff across membranes, and we said, oh, it's going to be really easy. We'll express the protein, we'll add some radiolabeled glycine, we'll see it go across the membrane. Pedro, who's in the front row, and I, who's very good at this kind of stuff, tried this for about a year, and so we said, well, that's not going to work. So then Pedro came up with some very bright ideas, along with Noelia, a graduate student in the lab, about using yeast genetic methods to try and actually prove that HEM25 is essential to get glycine into the mitochondria. And the process was to essentially design a yeast cell such that if it can't get glycine into the mitochondria, to actually do what glycine needs to do in the mitochondria, it'll get very sick. And so how do you go about doing this? So this is essentially what we were saying. We want, if we can't get glycine into the mitochondria, how do we make a sick yeast cell? Well, what's glycine used for besides heme synthesis? Glycine is used to make 1 carbon units, also known as folate. The only way glycine can make 1 carbon units is to get it into the mitochondria before it can make these 1 carbon units. The cell has a second way to make 1 carbon units, and that's from serine in the cytoplasm. If we inactivate the capacity of the cell to actually make 1 carbon units in the cytoplasm by inactivating its capacity to make serine, then the only way it can make 1 carbon units, and these are essential for life, is to take glycine imported into the mitochondria and make 1 carbon units this way. So we had a single mutant here, a single mutant here, and then the double mutant. And the double mutant is the presence of glycine or not, should or should not be able to grow. That's essentially what you're asking, right? And so the experiment was done. You have a wild type cell. If you add serine, where you're bypassing that genetic defect, everything grows. And then decreasing concentrations of glycine. And these are just yeast cereal diluted on a plate. And here you're looking at when you inactivate HEM25, the SLC25 homolog, they grow fine. Serine 1, they grow fine. You inactivate both, and you can see at the lower concentrations of glycine, they grow very, very poor. So that means when you rely on getting glycine into the mitochondria to synthesize 1 carbon units and you inactivate HEM25, you no longer grow, hence you're not getting glycine into the mitochondria, is the take-home message from this slide. There was a second hypothesis to test whether or not HEM25 was a glycine transporter. Glycine can serve, because it's got a nitrogen group in it, as the sole nitrogen source for cells, but again, only if it's imported into the mitochondria, because as part of the glycine cleavage system in the mitochondria, you end up making ammonia, and then the cells can use this as their sole source of nitrogen. So if you grow cells on a media where the only way they can get nitrogen is from glycine, but glycine has to go into the mitochondria, and you inactivate HEM25, you ask the same question. Now can they actually grow again? So here's a wild-type cell growing in the presence of glycine. Inactivate HEM25, they're clearly growing slower. If you inactivate the glycine cleavage system, they clearly cannot grow at all. The fact that there's a partial phenotype here and a partial but not complete loss of growth here implies that although HEM25 can import glycine into the mitochondria, because the phenotype's partial, it's probably not the sole glycine importer, but it's a very important glycine importer. To further hammer home the fact that HEM25 or SLC25A38 is a glycine transporter, yeast can make glycine through 3 routes de novo. These SHM1 and SHM2 proteins, and these are the human versions here, orphan Gly1. These 2 processes take place in the cytoplasm. This process takes place in the mitochondria. And we ask, what's the de novo source of glycine for a yeast cell in general, and the de novo source of glycine in a yeast cell to make heme? So you just inactivate each of these genes singly, and you measure how much glycine is in a cell and how much heme is being made. And when we inactivated the Gly1 gene, there was very little glycine and almost no heme, whereas the other 2 de novo sources of glycine really did not contribute to heme synthesis very much. As Gly1 is in the cytoplasm, this is again consistent with SLC25A38 and 25 having to bring glycine into the mitochondria to then make heme. So we've got 3 different ways that are sort of genetic arguments that point to the fact that SLC25A38, the yeast homolog HEM25, import glycine into the mitochondria for subsequent synthesis of heme and hemoglobin. However, because all the phenotypes we saw were partial, they weren't complete loss of function, when we made yeast rely on importing glycine in the mitochondria for growth, they're probably not the sole importers of glycine. So there must be a second glycine transporter, either a lower abundance or lower affinity, that allows yeast to actually import glycine through a second mechanism. We're searching for this guy right now. So now we know the function of the gene. It's actually a glycine importer into the mitochondria. That's great. With this knowledge, can we find a better treatment or hopefully cure for SLC25A38 mediated congenital sideroblastic anemia, now that we know what the thing does? So congenital sideroblastic anemia, it's rare, about 1 in 50,000 people. As I said, there's 2 genes, ALAS2 and SLC25A38, that can cause it. ALAS2 patients can respond to pyridoxine, and that's because pyridoxal phosphate is a cofactor for this enzyme. So some of these mutations actually allow restoration of enzyme activity in the presence of pyridoxine. The remainder need blood transfusions. All SLC25A38 patients do not respond to pyridoxine, and they require lifelong red blood cell transfusions every 3 to 4 weeks, which is no fun. And that comes with iron overload. Both the disease itself can cause this. Blood transfusions itself also lead to this, even with the use of iron chelators. What happens with iron overload? Well, a bunch of your organs fill up with iron and they don't work very well, and of course your lifespan gets shortened significantly. So, the current treatment, transfusions, physician visits, the oral chelator, hospital admissions for complications, the annual total for treatment for a CSA patient comes in at about $60,000 a year in direct costs right now. So one, it doesn't work very well, it works okay, but not great. It's expensive, and it's not fun to get transfused every 3 weeks, right? So we use our little paradigm, establish a model organism, we've already got a yeast model, to then try and identify, using either high throughput genetics, high throughput drug screens, or occasionally didactic reasoning, the supercomputer thing attached to our necks, to try and identify a process by which we could find a potential drug target or potential therapy. So did this work for congenital sideroblastic anemia? Yes and no. The first approach we took was very simplistic. Now that we know this is a glycine transporter, but we also know there's a second glycine transporter that we don't know about. There were 3 different scenarios we thought might be able to restart the heme biosynthetic pathway when this gene was inactivated. The first was to add serine, and nobody knows how serine gets into a mitochondria, but if it does, could we add enough serine so that we could drive it to glycine to drive it through this pathway? The second was to add glycine, but in excess, such that we could overcome the partial phenotypes we saw, so that we could get glycine into the mitochondria and then drive the pathway. And then the third was to bypass this whole process completely and add a metabolite downstream of the pathway, 5-ALA, and just see if we could bypass this genetic defect and actually synthesize human hemoglobin. So basically we took a yeast cell where we'd inactivated Hem25, which are these 3 bars here, and added either glycine in excess, serine in excess, or 5-ALA in excess, and asked how much heme do we synthesize compared to a wild-type cell in the Hem25 knockout. And you can see when we added glycine in excess, not serine, but 5-ALA in excess, these 2 small molecules, glycine and 5-ALA, were able to bypass this genetic defect and restore heme levels to normal if you're a yeast cell. So yay, could we translate this into a higher model organism? So this was where we collaborated with Jason Berman and Sergey and Shelby in his lab, and they created a zebrafish model of this same disease where they inactivated SLC25A and B. It's duplicated into the fish genome, so you knock down both genes. And what they've done here is they've used a stain, O-dianicidine, which stands for hemoglobin itself, and they've asked, all right, when you inactivate these genes, well, you essentially get no hemoglobin. Not surprisingly, that's what you expect, the fish become anemic. You add excess glycine, really doesn't help much, which was dreadfully annoying. Same thing was saw when we added excess 5-ALA. We also did not see much hemoglobin compared to a wild-type fish. So our hypothesis clearly sucked, and we were stuck with this question. Why were we not able to cure the zebrafish when we knew we could restore heme synthesis if you're a yeast cell? So we thought, well, within the mitochondria, once glycine's in there, what else is it used for? And it's used to make, as we said, tetrahydrofolate, so 1 carbon units. Folate's very important, of course, for everything. The cell actually keeps its folate levels right around the level it's required for use by folate-requiring enzymes. And we all know if you don't get enough folate in your diet, especially pregnant mothers, it can lead to all sorts of problems, right? So folate deficiency in higher organisms is a really big problem. The thing is, if you're a yeast cell, these cells can make their own folate. So it's really not that big a problem for them. However, zebrafish, mice, humans cannot make their own folate. We have to actually eat it as a vitamin. So we said, well, perhaps that's the problem. We actually need to add glycine to restart hemoglobin synthesis, and some of the enzymes required to make hemoglobin are folate dependent, so you need the combination of both to drive the entire process forward. And so this is your knocking out the SLC25A gene and staining for hemoglobin by itself. This is adding glycine, this is adding folate, and this is adding glycine plus folate compared to just a wild type of fish. And if you look at quantification of a bazillion fish, you can see this is what normal levels look like. This is where we get when we add glycine and folate compared to the non-liver fish. So by and large, we can restore the capacity of these fish now to actually synthesize hemoglobin, which is the major defect present in these patients, just by adding glycine plus folate at higher levels than you would normally have them in your diet. So what did we learn over the last, and this has been about 2 years' work on this project. We learned SLC 25A38 is a mitochondrial glycine transporter. Glycine and folate ameliorate the yeast and zebrafish model of congenital sideroblastic anemia. Because it's glycine and folate, these are actually dietary supplements. They're also used as drugs, but they've been around for so long that they're very, very safe. We called Health Canada, and they said, go ahead and put in a clinical trial application based on the evidence you have in hand. And if we're lucky, this could represent a potential low-cost breakthrough for the treatment of congenital sideroblastic anemia. So we're hoping to start the clinical trial within a year to see if this works. So that's one of our stories. Where am I in time?
Oh, you've got, 13 minutes left.
Perfect. Okay, good. So I'm going to give you a second example. This one's one that's kind of a little close to our hearts. There's a very nice girl, Tara Soller, who's helped us out from the very beginning of this project. She was a patient admitted to the IWK. She was diagnosed with this disease, familial exudative vitreoretinopathy. She helped us with the IGNITE launch in terms of the press event. Her and I and my mother also went to Ottawa to lobby the MPs and the senators to try and throw more money at orphan disease research because it's become a tractable problem to solve now versus the intractable problem it was a decade ago. And this is a video that Sue from Genome Atlantic put together when we first launched IGNITE to try and put the whole process in perspective of what we're trying to accomplish. Tara's completely blind.
However, right at the beginning, it was very devastating. You don't know what to expect, right? You don't know how you're going to raise a child with this disease.
The initial shock and devastation of it, you really have to cope. You know, you're left with what do we do? So. Consider ourselves, lucky that Tara did have a tiny bit of vision in one eye. We discovered, so she was able to work with that a little bit. But when she turned, I think it was around when she was 10 and a half or 11, the disease got active again. And when it got active, over a period of about a year to a year and a half, it just slowly took that vision that she had away.
It's younger, I thought the vision that I have is normal, because, I could see from, in my opinion, because, I could see where I was going, I could see people, and see different things, and I'm really lucky to have been able to see that, because, it's hard to explain color, and I'm just lucky that I got to see that before it was gone.
The research that they've done up to this point in finding the gene is really the first step. It's great to know where it comes from. Now, the big question is how do you treat it or prevent it from happening in other people.
Think about what I've gone through, and to think about that happening to other people and how other families are trying to figure out what to do, kind of thing. It's just kind of, it's not what is good, I guess. It's not right for that to happen. And it would just be nice to say, oh look, someone has FFPR, we can do something good.
So of course, Tara and her mother have been dreadfully helpful as far as the project goes. One of the genes that causes fever was discovered here in Nova Scotia through patients admitted to the IWK again. Fever is an inherited blinding disorder. The symptoms start in early childhood or your teenage years. There's no treatment specific to the disease right now. You can treat some complications of the disease using laser, retina detachment surgery, or possibly Avastin, although it doesn't seem to work very well. So it's still pretty intractable in terms of trying to cure this disease, as Tara's case clearly illustrates. And all current treatments essentially have poor outcomes right now. So is this something we could do something about? Well, there's been 4 genes, 5 recently, but 4 traditional genes known to cause fever. Frizzle-4 was discovered here in Nova Scotia, and 3 others have been added to that. And they're all part of the same signaling complex. So here's Norin is one disease, LRP5 is the other gene, T-span12 is one, and Frizzle-4. And Norin is a ligand that binds Frizzle-4, which then associates with LRP5 and T-span12 to set off the signaling cascade that results in changes in gene expression that allow the vasculature in your eye to actually develop. So if this process is off, there's no change in gene expression, and the vasculature of your eye doesn't develop. Without any vasculature in your eye, you can get no oxygen to the periphery of your eye, and of course it dies and you go blind. So we had some molecular information, and we knew a lot about the phenotype of fever. And one of the nice things about these genome candidate grants is they are a fair amount of money, so it does allow you to occasionally take a bit of an expensive flyer as far as the disease goes. And this was one of those flyers where you said, you know, this is probably far-fetched, but it's worth doing if it works because it'll be very transformative. So I've worked on lipids and lipid signaling for 20-odd years now and keep up with the literature. And there's a class of receptors that bind to a lipid called sphingosine-1 phosphate. Not too important, other than these 3 receptors are all found in endothelial cells, which are the problem in the vasculature in terms of trying to actually get the vasculature to develop properly. 2 of these are actually involved in driving vascular migration, and one of these, the sphingosine-1 phosphate receptor 2, when it's on, it inhibits vascular migration. Our thought process was if we inhibit the inhibitor of vascular migration, that should allow migration to now restart. And that might help revascularize the eye. So the other nice thing about the sphingosine-1 phosphate receptor, if you're looking for a drug target, is if you're going to shut something off, you want to make sure it's not going to actually cause more harm than good. And the nice thing about these sphingosine-1 phosphate receptor knockout mice, which exist, is they biologically develop hormone. So all that looked good. What's the experiment that we're going to do? We're actually going to take these knockout mice, and they've actually made knockout mice for each of the genes that cause fever and make a double knockout mouse. So if you take a fever knockout mouse that cannot see, and then you inactivate in that same mouse this gene, and you can revascularize the eye, you can then get a drug that inhibits the protein encoded by this gene without a cure for fever. Right? So again, we went ahead to make the double knockout mice. This was work done by Mike No, and you can barely see this. Are there those lighting switches somewhere? All right, pretty dark. Anyway, so this is what a retina looks like, and this is the vasculature. In a wild-type mouse, if you inactivate one of the genes that causes fever, you can see there's a lot less vasculature. It's nowhere near getting to the periphery when it needs to get there. But if you inactivate the sphingosine-1 phosphate receptor gene in this mouse, vascularization is restored. So you can essentially bring vascularization back to normal in this mouse. So we inactivate our potential drug target, and it ameliorates fever in this mouse model by making the double knockouts to prove that this actually works, and the fact that the mice survive means that it would be a good drug target. We've performed an in silico direct cheminformatics drug discovery lab, which means if you know the protein structure, which we do for this, you can use computer-aided drug design to try and dream up some drugs, which we plan on testing. Also, pharmaceutical companies have looked at this class of receptors to treat agonists. So instead of turning it on, turning it off, turning it on to treat multiple sclerosis. So they may actually have drugs on their shelf that are antagonists for the same class of receptors. And we're visiting several drug companies over the next couple of months to talk to them about this and other IGNITE projects to see which compounds they have on their shelf that they know are safe that we could then try on our models to see if we can move forward with it. So this is another project that's been pretty successful. It's very difficult to do a high throughput screen of small molecules in mice. One, it's expensive. Two, it takes a long time. So in collaboration with Jason Berman's lab and Shelby and Sergey again, they knocked out the Frizzle-4 gene, which is one of the genes that causes fever. In zebrafish, you can see they get the same vascularization type of problem. So the zebrafish will allow us to then have a high throughput model to screen for small molecule drugs that are ameliorative and those that work and are not toxic to the fish. We can then move forward and test in the mouse models. So that's our plan of attack right now for this. So to summarize, orphan diseases collectively impose an enormous burden on individuals, families, and society. Genomics research promises a major impact upon these burdens, with diagnostics being the first major impact. Some of these therapies that we then can find for a subset of these orphan diseases could possibly be leveraged to more common diseases. An example is fever looks a lot like retinopathy of prematurity, which premature newborns get. There's a genetic link between fever and ROP, which makes you think of therapy for fever might work for the more common ROP. Beyond these good things, there are many ethical and social challenges exist that when you get your genome sequenced to try and diagnose things prognostically. And IGNITE is providing a platform to study the means to ameliorate many of these orphan diseases, both diagnose and ameliorate. And I'll give you a couple of examples of some projects thus far. These are all the people. As you can see, it's a ginormous list of people. It's scattered throughout the IWK, Capital District Health, and Dalhousie University. Those in red were the ones that participated in the projects that I outlined today. And of course, the people that helped fund the research, by and large, Genome Canada and Genome Atlantic, with input, sharing the pain from a lot of other funding institutions. And I'd like to thank you for your time.
So because it's close to 1 o'clock, I'm just going to make a few announcements before we do the question period, just because some people may need to leave at 1 o'clock. So for those of you who are collecting CME credits, you can get a CME accreditation form outside, fill out an evaluation form, and we want the evaluation forms to be filled by everybody because they're going to help us to determine what you like and don't like and make it better for next time. And then the next topic will be on June 4th. It's not going to be at the end of the month like it usually is. It's actually going to be on June 4th, and Ian McDonald, who is from the University of Alberta, will come and talk to us about his work on ocular gene therapy. He's starting a trial, actually, in people with choroideremia. And then so this will be the last one for the season. The first one on September 10th will be Paul Lasco, who is the scientific director of CIHR, and we will provide more details as we go along. Also, if people have missed previous seminars, you can find them on links to the videos from the website. And then there's one more thing outside on the way out. You'll see that there's some flyers on the session organized by the CORE, the Canadian Organization for Rare Diseases. There's going to be a forum organized in Halifax on June 6th. And all stakeholders are invited to attend. The idea is to strategize for rare diseases and personalized approaches to drug access. It'll include everybody, patients, healthcare providers, decision makers, and people from the industry. So anybody who is interested will be invited to attend. The exact location is yet to be confirmed, but we will send out a flyer very shortly as soon as we have all the information to all of you who are on our mailing list. Having said that, does anybody have questions?
Thank you, Chris. That was lovely to talk. I have a question regarding the sideroblastic anemia and the folic acid, or the folate. given that compounds to enter into the mitochondria need to be positive charged, the folate is both hydrophobic and not positive charged. How did you compensate or overcome? Did you conjugate it to something?
No, there is a folate transporter in the mitochondria that can transport folate into the mitochondria. So we just imagine that we're just saturating the transporter more than it normally is saturated as far as folate goes.
So how did you overcome the hydrophobicity of folate?
Oh, we didn't have, well, we made folic acid.
Okay, so you made converted to an acid.
Yeah. Morgan. I just hired Morgan, so it better be a nice question.
So you mentioned, you found this one gene that causes the same phenotype, same diseases as this other gene. I mean, is that occurring in the study so far with Ignite? I mean, are you seeing things where you thought it was this one gene already, or it seems to be maybe multiple genes? Not necessarily require multiple genes, but mutations in different genes.
Yeah, so for a lot of these rare diseases, what we're seeing is mutations in more than one gene will cause the same disease. And they're often within the same pathway, or they're on a parallel pathway that has a similar biological endpoint that will result in the disease. So that's one of the nice things for orphan diseases as well. It's not in every case, because we don't understand everything that goes on in a cell or organism yet. But most of the times we can piece together, once we know the functions of the proteins, or if they're already known even better, the molecular pathway or the defect that leads to the disease that then allows for some intelligent design when it comes to which model organism to make and which type of screen or didactic reasoning to use to then move forward for a drug target type of approach or a small molecule type of approach. But yeah, they usually end up in a complex. Like the fever genes, all four are in the same signaling complex. It's often the case. Spectacular, we can eat.
Well, thank you, Chris.